morning to all of you. I'm here at my university and I'm going to be studying again for my cell bio class. This is going to be chapter 2 of like some review questions here and there, so please sit back, relax, and let's get started. Alright, so in the background I'm going to be playing Trombone Concertino in E flat major by Maestro Henry 18. So which of the following covalent bonds is relatively nonpolar? So we have the bond between carbon and hydrogen, C to H. We have N to H or O to H. So N to H and O to H kind of have charge on them because of that electronegative oxygen. So let's go to C and H. Right, what chemical group is found in a nucleotide but not in a nucleoside? Alright, so nucleoside and tide. I think the phosphate group. In water, hydrogen bonds can form between which of the following? So it's the hydrogen atom of one water and the oxygen of another. So let's see which one this is. There we go. How do protein, nucleic acid, and polysaccharide molecules polymerase or grow in length? By condensation. Which term describes a fatty acid? Let's see, hydrophobic. But they also have a hydrophilic head of glycerol, so this could be amphipathic. Amphipathic means an area that's hydrophobic and an area that's hydrophilic, so let's pick that one. You have developed a promising anti-cancer agent, but it has poor solubility in water. Which of the following changes should improve water solubility? Okay, so we're looking for the addition of kind of a polar group, because those are the types of molecules that will bind well in water. So let's see. Adding a methyl, no, that's CH. Hydroxyl, maybe. So let's go with adding a hydroxyl, because that's a polar group. So is a carboxyl group. That's like a carboxylic acid. So let's pick those. You have those good oxygens there that have really nice electronegativity. So then we'll draw electrons unequally to themselves. So that creates a partial negative and a partial positive to whatever they're binding to. Alright, what type of bond is formed when two atoms share electrons? So sharing, that would be covalent. Which chemical group is found on all amino acids? Right, so we have a carboxylic acid and a nitrogen, as well as a side chain. The side chain will change, so do we have the nitrogen group? Let's see. No, we do not, so we only have the carboxylic acid group, so that has to be the correct answer. Which of the following does not contain sugars? Steroids, those are lipid, glycolipids. Of course, they have glyco, so that means sugar. Cellulose is a sugar. It's the structural sugar in plants. Glycoproteins, again, glyco means sugar, so that's a protein with a sugar attached. Nucleotides do have a sugar. They have deoxyribose or ribose, so I'm going with steroids. A cell's plasma membrane with a greater degree of saturated or unsaturated hydrocarbons will exhibit less fluidity at room temperature. Alright, let's see. Can't remember this one exactly, but I'm going to go with saturated, because a saturated lipid only has carbon and hydrogen bonds. You don't have any double bonds, which will create kinks. So it's more stable. If you stack up a bunch of these saturated fats, then you don't have a lot of wiggle room. Whereas if you had a double bond, you kind of get a kink, so that's unsaturated, because some of the carbons do not have all hydrogens attached to them. So you get these kinks in the double bonds, so that will make it kind of wonky and more fluid. So let's go with the less fluid option of saturated. Which bond term describes a covalent bond in which electrons are shared unequally? Alright, so polar or ionic? I think it's polar. Which four elements make up 96% of weight of living organisms? So we have carbon, phosphorus, sodium, hydrogen, carbon, hydrogen, nitrogen, and oxygen, carbon, O, Na, and H. I don't think any of the ones with sodium Then this one has calcium too. While calcium is important for the human body, I don't think it's very prevalent other than the bones. So it says also all living organisms. So let's just pick carbon, hydrogen, nitrogen, and oxygen. What type of bond links two polynucleotide chains to each other in a double helix of DNA? Alright, so the chains are linked by hydrogen bonds, but the backbone is phosphodiester, so let's pick the hydrogen bond. What reaction involving ATP releases a large amount of energy? It's the release of that terminal phosphate. There we go. Based on your knowledge of other biological macromolecules, what are reasonable assumptions about your macromolecule? Alright, so let's see. Monomers and molecule are connected to each other via hydrogen bonds. That seems like a good answer there. Let's see. That could be one. That's possible. It is synthesized via hydrolysis of monomers. That we know is incorrect because hydrolysis breaks things. We're trying to make something, not break something. So that one's not going to be it. It contains carbon-carbon bonds. Heck yeah, I think that one's going to be one of our answers. And then its cellular synthesis is directed by enzymes. Enzymes are prevalent in a lot of these synthesis reactions, so I'm going with C and D. Which statement is true of hydrophilic molecules? Right, so they're typically hydrocarbons, so nope, because those are hydrophobic. They don't have any partial charge, so water isn't going to really mess with them. Hydrophilic, they do mix with water really well, so might be it. Approximately how much does a mole of carbon atoms weigh? Okay, so 12 grams, because that's the molar ratio, the molar weight. Radioactive isotopes. Radioisotopes are elements are commonly used in biological experiments as tracers to follow and detect molecules of interest. For example, photosynthetic intermediates produced during carbon dioxide conversion to sugars were detected by exposing algae to carbon dioxide containing a radioactive form of carbon. The radioactive form could be rapidly detected in molecules produced by the algae during carbon fixation and sugar production. Why can radioisotopes substitute for non-radioactive isotopes of elements in experiments? Okay, so a radioisotope only has a difference in neutrons, but it's still the same element, so I'm going to look for that. There we go. Which type of covalent bond allows for rotation about the bound axes? So it's not the double, because those are stronger. It's not the triple, because those are even stronger bonds. Let's go to the single bond. Which of the following would most likely interact by forming an ionic bond? Okay, ionic. So it's like sodium. So sodium have Na plus and Cl minus. We have to look for something like that. So an anion and a cation. So magnesium and ATP. Which of the following atoms is most likely to participate in an ionic bond? Chlorine with an outer electron shell. 7 possible. Carbon doesn't really do ionic bonds. Phosphorus, we don't really see any ionic bonds in phosphorus either, so it must be chlorine. On the basis of weight, which is most abundant in a living bacterial cell? Okay, this one is going to always be water. Are covalent bonds in the cell rapidly broken? Alright, so let's see. Enzyme catalysis, no, I don't think so. Energetic molecular collisions, nope. Nope. And it must be by energetic molecular collisions with the water molecules, because we have to add water and hydrolysis to break bonds. What types of bonds link together the components of a ribosome? Alright, so let's see. We have van der Waals, we have hydrophobic, and we have ionic, so we need to see these. Both the types of bonds. There we go. You discover what you think is a new macromolecule in a bacterium isolated from a pond near where you live. The following questions ask you why you think it's a macromolecule and what properties you predict it has. What evidence may have led you to conclude that the molecule is a macromolecule? Alright, so composition shows carbon, oxygen, nitrogen, hydrogen, sulfur. That doesn't tell us it's a macromolecule, it just tells us it's made of those elements. The molecule is essential. Nope, because there are ions that aren't molecules, because ions are just a single atom. Those are also essential, but they're not molecules, so that one's incorrect. During centrifugation, the proposed macromolecule sediments as a single discrete band, so that means it's one molecule, so that could be it. Regarding acids, bases, and pH, which of the statements is true? Substances that release protons, so acids, when they dissolve in water, are termed acids, yep, and they result in a lower pH. I think that one's the correct answer because a lower pH means more acidic. What is not true of RNA? Contains a different sugar, that's true. Is usually single-stranded, that's true. Contains a different base, that's also true, so that means this fourth answer here is the correct one. Cannot, which is not true. So it can actually form base pairing with other nucleic acids. Which of these molecules would you least expect to easily dissolve in water? Alright, so we're looking for something that has a ton of hydrocarbons and no partial or ionic charges. 2-methylpropane, because that's a hydrocarbon. What type of reaction is the reverse of a condensation? So condensation is when we're building. Hydrolysis is when we're breaking. So let's pick hydrolysis. The bond type between sodium and chlorine anion, so sodium cation and chlorine anion, that is an ionic bond. The ultracentrifuge is an important piece of laboratory equipment frequently used by cell biologists. What are some applications for the ultracentrifuge? Choose one or more. Alright, so we could separate, different... we can't determine whether prokaryotic cells are bacteria or archaea with the ultracentrifuge. We can't do that. We could, let me think. Choose the sequence. No, we can't learn anything of the sequence, but we can separate different cellular organelles from each other based off of weight. Let's submit. Oh, we got one incorrect, so 97%. Let's see which one we got incorrect. We got... How are covalent bonds in the cell rapidly broken? By enzyme catalysis. Okay. So it's specific between protein and substrate. Alright, so not too bad on our first run here. Thanks so much for tuning in for this short study session on Chapter 2 in cell biology. Please be safe, do something nice or so, and I'll catch you guys later. Thank you so much for tuning in.